Hello, Jocelyn, Jackie, Isaiah, and I will be doing our disease presentation on Lesch-Nyhan syndrome. So what is Lesch-Nyhan syndrome? It was first discovered in 1964 by Michael Lesch and William Nyhan and a pair of brothers at Johns Hopkins who displayed an unusual set of symptoms. It is a condition characterized by multiple neurological and behavioral abnormalities, as well as the overproduction of uric acid. It is a disease that is mostly prevalent in males due to being an X-linked recessive genetic disorder, and has affected approximately 1 in every 380,000 people. Most people who are affected by Lesch-Nyhan syndrome do not tend to survive past the age of 20, and this is mostly due to renal failure. Signs and symptoms of Lesch-Nyhan syndrome tend to begin as early as 6 months of age when uric crystals form due to high uric acid levels in the body. And it's also characterized by blood in urine, urate stones in the kidneys, and various other symptoms in infants as early as 6 months of age. Behavioral signs tend to start to appear as early as 12 months of age, with involuntary rhythmic movements, repetitive movements, and a failure to reach developmental milestones such as crawling, walking, being able to sit up on their own. However, it is not until the late teens or early adulthood that those affected tend to experience recurring episodes of pain and swelling in the joints, and episodes tend to become more frequent with age. Various symptoms that are seen in most Lesch-Nyhan patients include gout, high blood acid uric levels, hemiplegia, hemiparesis, hypo or hypertonia, assistance with sitting, and they typically cannot walk. There's also the flailing of limbs and self-mutilation, which self-mutilation behaviors would tend to include finger biting and head banging. A few other symptoms include impaired kidney function and neurological impairment. Females typically do not show symptoms until they are older when they develop gout, and then from there it just becomes multiple episodes of various symptoms.
So currently there are 3 different ways you can diagnose an individual with Lesch-Nyhan. One way you can diagnose an individual is with an enzymatic diagnosis, which includes enzymasase. In this test, undetectable to low levels of the HPRT activity in either erythrocytes or fibroblast cells will indicate an individual with the disease. Another way to diagnose an individual is with a molecular diagnosis, which includes the Southern blotting technique. In this technique, mutations on the HPRT mRNA will be identified, and therefore the individual can be diagnosed with the disease. Another diagnosis that can be used is a biochemical diagnosis, and this tests for high concentrations of serum urate or urinary acid creatine ratio. Most individuals with the disease will live well until their 20s, and it is possible for them to live past their 20s into their 30s with the help of the allopurinol treatment. Most of these individuals will be confined to a wheelchair, and most do express self-mutilation or self-injurious behavior. So because of this, they do have to be monitored by another individual to prevent them from injuring themselves. And then currently there are 3 different treatments available. One treatment is the allopurinol treatment, and this treatment inhibits xanthine and hypoxanthine from converting into uric acid. And because of this, uric acid concentrations are then reduced, which also reduces symptoms associated due to high uric acid concentrations. Another treatment that is currently used are benzodiazepines and gamma-aminobutyric acid inhibitors, and these are muscle relaxers. This helps mainly with the motor symptoms such as managing spasticity and dystonia in individuals. And also, physical restraints and guards are used to prevent self-mutilation behavior, so elbow restraints will be used to keep the fingers away from the mouth, as well as dental guards to prevent them from biting their gums and their fingers. Currently, there are experimental treatments available. One treatment includes the use of gabapentin. This use increases GABA levels, which increases serotonin levels, which decreases self-injurious behaviors. Currently, there are no side effects to the drug, but it is not approved by the Food and Drug Administration for use in children 12 or under. Another experiment is the botulinum toxin. This toxin is injected into the bilateral masseters, and it will affect the central and peripheral nervous systems, which will therefore result in a decreased self-mutilation behavior. Both these treatments aren't currently used because their long-term effectiveness and safeness has not been proven.
Now that we've discussed the signs and symptoms of Lesch-Nyhan, it is important to understand why this occurs and where it occurs. So Lesch-Nyhan occurs due to mutations on the HRPT1 gene, as it was discussed earlier. This gene is located on the Q arm of an X chromosome. Specifically, it's located on the region Q2627. Now, this enzyme is important because it contains a functional messenger RNA transcript that encodes for the enzyme HGPRT. This enzyme, as we'll further discuss later, is important and is responsible for purine metabolism. According to multiple studies, including Fu et al. 2014, there has been no specific mutation or specific location on this gene that causes this disorder. There are multiple different severities of the disorder, and those severities are due to different mutations on this gene. Now, going over to our next slide, we're going to further discuss the enzyme that this gene codes for. This enzyme is called hypoxanthine guanine phosphoribosyltransferase, HGPRT. Now, as we discussed earlier, it is responsible for purine metabolism. And the reason why it's important for purine metabolism is that it recycles chains of RNA and DNA, specifically the purines of these RNA and DNA recycled chains, and it produces nucleotides. So it does this by binding purine to sugars. As we can see on the biochemical pathway here, specifically it's a purine metabolism pathway. We see that that in the salvage pathway of this purine metabolism, HGPRT is responsible for breaking down P-ribose PP into GMP and IMP, which IMP further becomes AMP. Now, this is important for purine metabolism because HGPRT is producing energy and through GMP and AMP, which will further become GTP and ATP. And this energy is important for DNA synthesis, and it's important for protein synthesis later. Now, what's even more important about HGPRT and how it relates to Lesch-Nyhan is that through this purine synthesis and through the breakdown of these purines, or the purine degradation, it is lessening the amount of guanine and hypoxanthine that becomes into uric acid. So it helps prevent and lessens the amount of the toxic products, such as ammonium and uric acid. Now, uric acid and the buildup of uric acid is what causes the symptoms of Lesch-Nyhan. As I've discussed earlier, there is no specific mutation that causes Lesch-Nyhan. There's various different mutations that can occur on the gene that can result in the same type of result. Through the study of Fu et al. Specifically, I did notice some patterns they found their own conclusions on how mutations in the genotypes specifically affected the phenotype of Lesch-Nyhan. So for mild symptoms, they noticed that duplication mutations tended to produce milder symptoms of Lesch-Nyhan, which would be gout, it would be uric acid buildup in the joints and in the limbs. And then they also noticed that severe symptoms, such as severe symptoms, were caused due to deletion mutations or missense mutations produced by were producing severe symptoms. According to the study, these mutations and the types of mutations produce these types of phenotypes due to how they affected the enzyme. So mutations that caused a total loss of enzymatic activity of the HGPRT affected and produced more severe phenotypes, while mutations that allowed some enzymatic activity and allowed complete activity of the enzyme produce a lot more milder symptoms.
For the proposed experiment portion of our presentation, we will be observing the relationship between uric acid and dopamine levels associated with Lesch-Nyhan syndrome, because many scientists who have studied LNS patients have found an association between increased uric acid levels and decreased levels of dopamine in the brain, but no one really knows why this occurs. So our goal is to determine if uric acid plays a direct role in decreasing the levels of dopamine. And our hypothesis that we came up with is that uric acid inhibits the dopaminergic pathway by blocking the release of dopamine into the brain. The setup for our experiment is as follows. We will be experimenting on 10 rat embryos and allowing them to fully develop into rats. The rats would be divided into 2 groups of 5, one group being the control and the other one being genetically induced with LNS. And this can be done through the genetic manipulation of the rat embryos, which is called mutagenesis. And the gene that we're specifically manipulating is the HPRT gene, which is a characteristic component of development of LNS. In order to track the location of uric acid and dopamine in the brains of the rats, we will be using radioactive tags so that they can be visualized. We will visualize the location of these 2 molecules through positron emission tomography, which is a tool used to visualize metabolic processes within live cells. And we will be taking these scans and comparing the scans that we got from the control group with the scans that we get from the genetically induced group to see if there's a difference in the location of dopamine and uric acid. Given our procedures and the potential tomography scans from the proposed experiment, the scans would show a localization of uric acid and dopamine within the cells. And this could mean that uric acid would potentially degrade or inactivate dopamine and prevent it from being able to be secreted, or it degrades the vesicles responsible for transport of this signal, so it would not be able to leave the cell. Although this does not completely determine if uric acid's role, it would seem to play a part in the prevention of dopamine secretion. That concludes our presentation. Here is our works cited pages. And that's it.